Dr. Kuppermann, thanks so much for meeting with us today at the Retina World Congress. You had a presentation earlier called The Role of Mitochondrial Genetics on AMD. Can you outline your presentation?
Sure, this is some very exciting work that we're doing in my laboratory. I'm a professor at the University of California, Irvine, the Gavin Herbert Eye Institute. So I have a busy clinical practice and train residents and fellows, but I also have an active laboratory that I share with Professor Christina Kenny. And there what we're doing, we have cells in culture that we've been developing for a long time. These are commercialized cell lines. We've run a lot of assays looking at gene expression in these retinal cell lines. But what we've done more recently and what we're studying now is that we're looking at the role of mitochondrial genetics on the expression of the nuclear genes, so that the mitochondria, the organelles within each cell in our body that are the power plants of those cells, they have their own genome. They have their own small circular genes, and those genes may influence the expression of the double helix genes, the 25,000 genes per cell that we have inside the nucleus. So what we do in our lab is I have patients that we've characterized their AMD, how they've responded, what's been the course of their therapy, have they been good responders, have they developed geographic atrophy, what's happened with them. Then we draw their blood and we take their plasma, which are loaded with mitochondria. We deplete the cell culture lines we have of immortalized retinal cells, and we deplete them with ethidium bromide of their mitochondria, and then infuse the mitochondria from the patient into those immortalized cell lines. So now we have these hybrid cells, cytoplasmic hybrids, which are dubbed cybrids, and those now have the mitochondrial genes of a patient with active disease, and yet they're in a retinal cell line that has immortalized cell line nuclear genes. And then we run various assays and look to see what's expressed. So reactive oxygen species, that would be a manifestation of progressive oxidative stress that would be potentially tell us whether that patient was likely to develop geographic atrophy or not. Then we also run VEGF-A to see if that's upregulated or downregulated in response to an anti-VEGF injection. You would think that if you give anti-VEGF, you should see downregulation, but maybe not always. We also look at HIF-1 alpha, which is another agent that's important in the angiogenic cascade. What I showed in my talk were two patients in particular that we now are collecting this sort of library of patients and library of cell lines. So the first patient had good response to Lucentis, but developed geographic atrophy. And that patient showed increased reactive oxygen species in the lab, in the cell line of their cybrids, and that could explain why they developed geographic atrophy, but had downregulation of VEGF, they had a very good response. So that was actually a good VEGF response. Another patient, for example, needed a lot of injections. It was to Lucentis as well in this case, and it needed a lot of injections of Lucentis, was always reactivating, never developed geographic atrophy, and there was no real change in the reactive oxygen species, but had upregulation of their VEGF, which you would expect the opposite. So that patient in that case was not that sensitive to Lucentis, and one would consider changing drugs to consider another therapy, and we have not yet done that, but that's the sort of approach we're taking to personalized medicine is our hope that we'll be able to get there. The take-home message is that not all patients necessarily respond the same to all the drugs, and there may be profiles that we can identify from their mitochondrial genes that may help guide us into our therapeutic pathway. But again, that's personalized medicine that's still in the future. We're developing it now. We're not there yet, but we hope to be there soon.